in the team of Lucy and Jessica, the in-depth team dedicated to imaging neurodevelopmental phenotypes. That's the name of the team. And I will be talking again because it's been also a topic yesterday with the wonderful talk of Catherine about fetal alcohol spectrum disorder, but reminding you that this is a disease. It's called a disorder, but it's a stupid way of calling things. It's embryofetopathy alcoholic. It's the disorder, the disease, the developmental disease caused by alcohol exposure, as if you had a genetic mutation. And the point will be more to illustrate the fact that even with group effect, we can try, if we have a question, a precise question to ask, we can try to go from the group to the individual significance and the individual clinical significance. And this will be an example because first, the group is not so large, though here, Edouard said my group are small, but this is an even smaller group because it's 220 subjects, which is small. But the question is the same. With that group, with control and patients, a bit more control than patients, would I be able to have something relevant for clinic? And it's something where I think we've improved a little bit things because we had a very precise question, not such a big question as would I be able to do the diagnosis in the general population just with brain imaging? We had a very low level question, but it's perhaps also a point in the discussion, even in big data sets methodology, perhaps try to have specific question, and that was also perhaps the point of Ghislaine. So I have no conflict of interest, and just one second to thank the financing, the funding, and the association Vivre avec le staff for families, and reminding you that fetal alcohol spectrum disorder has been fetal alcohol syndrome, and the consequences of alcohol on development have been described in France a long time ago, but after French people stopped to be a believer on the subject, even if some in some places, some very important people such as Philippe Then, kept le flambeau on the matter in France. It has been described as a syndrome, meaning a bunch of clinical features that helps you to make the diagnosis of the disease by American people. And a few years later, a syndrome is an association of symptoms that describe the commonality of the pathophysiology in the patient, called fetal alcohol syndrome, with several impairments that I will remind you of. The point is that in that disease, there are two things that are of great interest. It is a cause of very complex and sometimes severe neurodevelopmental disorder, associating many of the categories that we use in clinic to describe the children, from intellectual disability to ADHD to coordination disorder, because they all have the criteria of that. The cause is alcohol, but they meet the criteria of the disorder. And they also have a specificity, which is brain growth impairment. They got smaller brain than the general population because alcohol is toxic of brain growth, as you've already seen with the talk of Catherine. It is a health problem because a public health problem because the prevalence is not low. It's not a rare disease. It's up to 1 birth per 1,000. And for the non-syndromic forms, it's perhaps 5 times higher. Fetal alcohol syndrome is the key that the clinician used to make the etiological link between alcohol exposure and the cognitive and behavioral phenotype that we call a neurodevelopmental disorder. That's the difference between the disease and the disorder. And the key for the diagnosis in clinics is to have a fetal alcohol syndrome, a malformative syndrome. And the important specific features in clinics is not the growth impairment, it's not even the brain growth impairment, it's the facial features, because they are specific. It's the same as with the Down syndrome, the fact that you have a neurodevelopmental disorder because you have 3 chromosome 21. Before genetics, the diagnoses were done on the face of the children. It's exactly the same. The syndrome, the physical syndrome helps you to make the diagnosis of the disease. The point is that there are 5 more there are 5 more children with fetal alcohol consequences that do not have the fetal alcohol syndrome. I will not elaborate on that. There are a huge amount of epidemiological data to show that. And for those children, the diagnosis is difficult. The diagnosis of the disease, the fact that it is alcohol that causes the disorders. So we are in need of markers to help the clinician make the diagnosis, which is the specificity of that disease. I can't have a blood sample and send it to the geneticist. They won't give me the diagnosis. I will do it because I need to exclude many genetic disorders that may cause many genetic diseases that may cause those neurodevelopmental disorders, but it will not give me the positive diagnosis. And we may need also markers for the prognosis and to understand the pathophysiology, of course. But there is a very basic question, clinically speaking, is it I am able, do I able to help for the etiological diagnosis when there is no fetal alcohol syndrome? So of course we turned to the brain because the brain is affected by alcohol exposure during the whole pregnancy. The face is probably affected during the early pregnancy, but the brain is very affected because those children have neurodevelopmental disorders that are very severe. They can brain growth impairment. So we expect the brain to give us more information even than the face. But if we look at the radiological defects, they are rare and they are very subjective. It's difficult to describe them. If you look to partial agenesis of the corpus callosum in that population, there is a slight increase, but there is a slight increase in general in the population of neurodevelopmental disorder. So it's very unusual. You can use it for the diagnosis. It's useless. To a point that in the guidelines, the official guidelines of the diagnosis, if you have microcephaly, it may help a bit the diagnosis, but any kind of disruption in the brain would help a little bit also, but add no specificity to the diagnosis. And after 25 years of computational neuroimaging, even if I take only the object-based morphology, well, no breakthrough yet to help for the diagnosis. I would go into detail. It's not because nobody cares, and it's not because there's no interesting studies. There have been very interesting studies in the field. Even, for instance, the impairment of the cerebellum has been described by Elizabeth Sowell 30 years ago now. And it was a very, very iconic first study because she really saw something that we struggled to show again. So things have been shown, but the studies are very small. The population are very heterogeneous and sometimes used several times. So I told you that in the bunch of study, we had something like 150 really phenotype fetal alcohol syndrome, which is very, very small because the population are used again and again. So it's not such a big population. But there is also another problem to explain why we don't reach significance at the clinical level is that nobody tried. Actually, all those studies were designed to find significant group effects, mean effects. And in the end, everybody described its mean effects, but nobody tried to answer really a clinical question. I think it's important because it shows that if you want to go into translation, you've got at some point to choose a clinical question. If you don't, you will increase the knowledge, which is very, very important indeed. I don't say the contrary. But if you don't try to address really clinical question, it will last a very long time before it really happens. this study by Little and Boudieu was very interesting because they first used a classifier in the end, but they did not use the classifier to specify the children, but to specify the region in the brain, which region is the more important to classify well. But in the end, they did not try to answer a clinical question, but a pathophysiological question. So we looked ourselves on the matter with a very now quite old preliminary study with Pauline Garzon, and we wondered whether there would be spatial heterogeneity in the brain growth restriction of that disorder, and this disease, the fetal alcohol disease. And the point at that time is that we showed that in our population from 8 to 18 years old, the effect of age was very small. And we had a small sample by that time. So we thought, yes, don't go into very complicated model with age effect, etc. Just look at the way the growth, the size effect is affect each region of the brain. So go into only scaling analysis, not growth analysis, not multifactorial analysis, but just scaling analysis. And to do so, as you've already heard, many studies just say, well, I have a size effect, so I make a ratio. I divide everything by the size of the brain, and I look if the ratio change. But the ratio change in the general population. If you take that example here, for instance, the cerebellum in the brain, if you take the proportion of cerebellum, the smaller brain, and you compare to the larger brain in the general population, the ratio change. So what would you do with that if you do not really study the continuous change of the proportion in the brain with size in a population where your patient cannot be paired with your control in terms of size? If you don't have a proper biomathematical model to project the proportion in the general population in that virtual area where there is no controls, you would use just a proportion model and you will miss the effect. So we try to implement nonlinear model of scaling and to see what would be the difference in scaling between patient and control, and what would be the patient compared to the theoretical control that would be their control if they had people in that size range. We did that in a very small population of fetal alcohol syndrome, and basically what we found is that the regions of the brain, this very raw segmentation, had an impact of the reduction that were very different if compared to the scaling observed in modeled in the control population. These deep regions, cerebellum and deep brain, would be very, very undersized, underscaled, and some regions would be, they're smaller than expected, but not as smaller as expected, a little bit protected. And this would be good markers. They would be allometric markers of the disease in normative analysis, but not normative growth analysis, but normative scaling analysis. And well, this was a very small sample. We struggled a bit to publish it and we stopped, but we tried it again, and you've got a poster of Jeremy in the hall showing that actually there is an excess of children with too oversized temporal region, and of course a smaller cerebellar region, for instance. So what we try to do in that situation is to learn from the fetal alcohol syndrome population, a fetal alcohol syndrome recurrent brain phenotype, and to see if it can extend to some children without a fully positive diagnosis, a kind of signature of the disease that we learn in the population where the diagnosis is sure, to see if we can catch some children with a non-specific diagnosis. We harmonized, we choose a few regions, not to be short in terms of statistical power. We harmonized to limit the effect of sites, because in our control we've got several sites. We residualize some small marginal effect if they were present, such as age, and then we go into one factorial analysis where two factors, the disorder and the disease and the effect of size. Then we try to see if there was a distance to typical scaling and an excess of children in the abnormal zone of the 10th to 90th centile of the control population. With that, we try to teach classifiers, even very simple ones, just additional additive classifiers, or more complex one, logistic one, to learn the signature in FAST and see if there were FAST-like brain in non-syndromic FAST-D. The population I will go very quick like that is the one it's research on health data. It was not an experimental data set. So we take advantage of the fact that in Robert Debré Hospital, with the help of the radiological service and the psychiatric service, we built a bunch of sequences that were both useful for clinic and for research. And we add the authorization, the ethical authorization to perform those this research on medical data. Very quickly, if you're interested in the phenotype of those patients, you've got the poster of Elliot Gerdreux on their cognitive heterogeneity and intellectual functioning. But I will just move to the fact that we had in the end 90 FASD with 60% of fetal alcohol syndrome and 125 typically developing children, with a bunch of them with exactly the same sequence, which is very useful if you want to make proper harmonization of the data and don't mess up your control population. We did with Justine Fraise a first study with very easy and handy, ready to use measurements for the clinicians, measuring the size of the brain with the reference area of the brain, measuring thickness of the corpus callosum, height of the vermis, and semi-quantitative assessment of this dysgenesia of the vermis that you already heard about yesterday. And we tried to see whether there were normative growth impairments. And there was a normative growth impairment of the brain size. But after, we thought normative growth impairment is almost all measurements because the brain is smaller, so the subpart of the brain are smaller. And with scaling a normative analysis, we saw only a recurrent callosal isthmus thinning and a recurrent smaller cerebellar vermis, with a bunch of them, small or not small, had dysgenesis of the foliation. We try to see if we can just have an additive information by showing that three abnormalities were quite frequent in both the fetal alcohol syndrome and the non-syndromic population, but not seen in the control population. And you missed the third line. I don't know why, but with three abnormalities, it is even more specific. And we try to propose to the community that these recurring abnormal scaling anomalies could be added to the clinical decision tree of the diagnosis, to say that 15 to 20% of the non-syndromic, they have such a brain that look like fetal alcohol syndrome brain, that they may have a more specific diagnosis than just, we don't know, you've been exposed and you've got the disorder, you got disorders. We try to replicate our data on the corpus callosum with a new technique of measurement of thickness profile that we developed with Julien Lefebvre, that you saw yesterday during the PhD of Justine, to have an object-based analysis of the corpus callosum from just the automatic measurement of the thickness, the singular thicknesses of the genu, the isthmus, and the splenium. Just one point to tell you that we found again the thinning of the recurrent thinning of the isthmus, but with that more systematic and probably less biased measurement, we found a recurrent thinning of the splenium and a little bit shorter that corpus callosum that we fade with the manual measurements. quickly, we developed also with the colleagues of Marseille and a segmentation of the corpus callosum to analyze the surface of segments. I won't go into the detail on the method because I don't have time and it was not the point, but it's a new method that could be interested in other developmental disorder or with involvement of the corpus callosum. And in a model that is more a multilinear model here, because we don't have enough controls to make really proper normative modeling, we showed that there was too small periismic regions, and that this could translate into a biomarker by performing normative analysis on the residual of the linear multifactorial model. We turn to the cerebellum, and there we try to see whether that reduction of volume of the cerebellum was homogeneous or not in normative scaling analysis again, but in the cerebellum. I go quick here because I'm short of time, but you see that the cerebellum is deeply affected in normative scaling analysis. It is really too small for the size of the brain, but it is not the case of all the parts of the cerebellum. The anterior parts are very, very small, in blue here, but the lateral and posterior parts, the ones that are the more involved in the cognitive networks, they are even a little bit preserved for the size of the brain and the cerebellum. They're affected, but they are much less affected than the motor regions of the anterior cerebellum. But more interestingly, for the point of the talk today, we trained classifiers with either just the information of the small brain and smaller cerebellum, or with the small brain and the whole scaling vectors of the segmentation of the cerebellum. And you see that it improves the way you can distinguish between FAS and controls. And if you set up the specificity of your classifier very high, 97% of specificity, which explains why you classify correctly only 60% of the FAS, then you classify in FAS-like 30% of the non-syndromic forms. So in that information, doing this type of scaling analysis and normative analysis, you end up with a thing that can help you to specify the diagnosis in the non-syndromic form. Very quickly, because you've got the poster, this is exactly the same methodology and aiming, not with cerebellum, but with the deep gray matter that is really impaired in that population. There is a deep undersizing of the deep gray matter, but it predominates on the caudate, for instance. And here again, you've got a gradient in severity, and it can help to classify the patient just on the morphological signature. There is difficulties because here we've got not a small effect of the segmentation method. So it's something that we can discuss and that you can discuss at the break with Elliot, with his poster. So in the end, is that all? It's all for the moment, because what we want to do now is to combine the whole brain analysis. It's not a whole brain because we've got only small segmentation. It's not over segmented, but it's adapted to the power that we've got in the series to see if we can do better than only with cerebellar or deep gray matter features. And of course, we want to know if those normative scaling markers could be useful to correlate with some part of the cognitive phenotypes, to be useful not only for diagnostic improvement, but perhaps for prognostic improvement. These are the people that have been very pleased to work with during the past 7 years of those work, even in our team and collaborators. And I thank you all for your attention.